Welcome to Tell Us What You Know, a series from Drug Discovery News where we ask experts to unpack some of the most present topics in science and research today. Omics research is evolving faster than ever, and new advances in mass spectrometry are unlocking deeper insights into proteomics, lipidomics, and metabolomics. Today we're joined by Paul Baker from SciEx. He's going to tell us how new mass spectrometry systems are addressing challenges in translational and pharmaceutical research, and how these innovations are reshaping the future of multiomics. Let's get into it. Hi, Paul. Welcome to the conversation today. So first of all, can you tell us where mass spectrometry fit into today's omics landscape, and how's your team at SciEx responding to some of the challenges?
Yeah, I think one of the most important powers of omics analysis is that it helps to establish a baseline of protein, metabolite, lipids, and other indices of metabolism in whatever sample you're interested in. And when you compare that to a non-control sample, it can help you identify molecules that may be related to certain disease states or metabolic disturbances. So in essence, it really helps to drive hypothesis formation. And it sets the stage for potentially more targeted quantitative work in the future. Now, how does today's mass spectrometry fit into omics analysis? I think that there's two limitations typically for omics analysis, and that's sensitivity and speed, right? So there's a term called coverage, and coverage relates to how many molecules within a sample are you actually able to detect, to process, to potentially quantitate, right? And typically, high resolution instruments have not been as sensitive as triple quadrupoles. So one thing that we're excited about at SciEx is the advent of the Xenotoph series of instruments, specifically the 7600 plus and the 8600. And what they did with this instrument is they tried to mitigate the typical low sensitivity that is experienced using, say, a QTOF instrument. And they did that by creating this part of hardware called the Xenotrap. And essentially what it does is it allows you to accumulate ions while ions are then flying through the time-of-flight chamber. It seems pretty straightforward, but actually in practice it was quite difficult to implement. But what that does is that in a typical QTOF instrument or a traditional QTOF instrument, ions are continuously entering the mass spec. At some point you have to shut the gate to the pusher and allow a population of ions to then fly through the time-of-flight chamber, right? During that time, the ions that are coming into the mass spec are wasted. So it comes. There's a term called duty cycle in response to a QTOF. The duty cycle is a number that represents the percentage of ions that enter the mass spec and actually hit the detector. And for most QTOF instruments, that's around 5 to 20%. So you're losing 95 to 80% of your sample to vacuum. Now, the Xenotrap allows us to trap ions while ions are being processed through the time-of-flight chamber, which brings that duty cycle up to about 90 to 95%. So right off the bat, the 7600 system and the 7600 plus system boosted sensitivity about tenfold compared to traditional TOFs. The 8600 with a much more advanced ion rail at the beginning of the front of the instrument now allows us to get 20 to 30 times more sensitivity, especially in the small molecule regions. So that's one way in which we're addressing the needs of the omics community is sensitivity. And when you have more sensitivity, you're going to get better coverage. And speed's also an issue. So you can have all the sensitivity you want, but if you can only process a particular data point, 5 a second or 10 a second, you're going to miss things. So the speed's important too, that you've got to constantly sample the ion stream coming through. And the Xenotoph has a detector which is capable of measuring as many as 133 MSMS per second. So those are the two ways in which they develop the hardware to try and address the needs of the omics community, which is to provide speed and sensitivity so that your coverage is actually increased. And that the more coverage, the more you know about your sample.
Great. So what actual advantages does that bring, and how does it translate into real clinical or translational outcomes?
Yeah, so as I mentioned, sensitivity and speed are going to improve coverage. The other aspect that's related to speed is the ability to do unique experiments, right? So the traditional discovery experiments are called DDA, or data dependent acquisitions. And that's where you essentially take a snapshot of what's coming into the mass spectrometer. And perhaps you see 100 peaks. You take the top 5, 10, 15, 20, maybe 50 peaks, and you do MSMS on those. And then you continue the cycle. There's an alternative method called data independent analysis, or DIA. SciEx actually pioneered the aspect of swath, the method of swath, or the workflow of swath, which allows you to not only generate data that can provide qualitative information, but it does it rapidly enough that you can actually quantitate simultaneously. So that simultaneous quantitation, qualification, or discovery is a really powerful aspect. Until recently, that's been really limited to the large molecule omics space, like proteomics. However, we've actually, with the Xenotoph instruments, we're actually able to use these very powerful tools for small molecule omics. One of the challenges with swath and small molecule omics is the fact that the window size is usually too large, and that you can't ascribe a precursor mass to the fragmentation pattern. And that's related to the field of lipidomics and metabolomics, where we don't have as many unique fragments as we would get with proteomics. There's a new workflow out called ZT scan, which rather than taking little blocks of the Q1 window, it actually scans through that. And so we can actually convert the data or deconvolute the data to actually identify the Q1 mass of that molecule. And it's exciting. It's fairly new, but it's, I think, really going to drive the ability to, in a single experiment, quantitate how much you have and discover what you've got.
And now let's get into the applications. So I'm wondering, how can advanced mass spectrometry you just mentioned support different research areas? Are there any applications or workflows where you've seen a strong impact?
Well, I'm going to be biased in my answer because I am a lipid biochemist, but I'll be quite honest. I think it's probably the biggest advance in a commercially released mass spectrometer for the field of lipidomics. And that lies in this new or complementary fragmentation mode called electron activated dissociation. So EAD is really kind of taking the small molecule omics world by storm, because what it allows us to do is really make up for the fact that there are so few fragments that are generated during CID. So collision-induced dissociation is a thermal fragmentation event. Typically, there are very few fragments and very few structurally diagnostic fragments that are generated during that analysis, in particular for lipids. However, with EAD, it's much broader in terms of fragmentation. The number of fragments is usually between 15 to 20 times more than we see with CID. And each one of those fragments actually has information about the structure of the molecule. So most mass spectrometers out on the market today can identify lipids down to what we call the fatty acid level, where we can say it's a PC molecule and it has 2 fatty acids. We know what the fatty acids are, but we don't know where they're at, and we don't know anything about the double bonds or the stereochemistry. And it turns out, with the electron activated dissociation paired with that boost in sensitivity we get with the Xenotrap, we're now able to determine the exact structure, complete structural characterization of lipids, in about 30 milliseconds. So it's really exciting in that regard. I think that tool is very valuable, and I think it turns out it's, based on some recent conferences I've been to, really appears to be capturing the imagination of people. And in terms of the Xenotoph, that's really exciting. That's a unique feature of our instruments, and it really is kind of causing a renaissance in the lipidomics field in terms of how we're able to report data. We're getting more coverage because now we actually can look at molecules in a very specific way, distinguish isomers in metabolomics and lipidomics and in proteomics.
So as we discussed in the beginning, omics research is evolving really fast, and more researchers are looking into incorporate omics level research into their mass spectrometry workflows. Do you have any advice for them?
Yeah, start slow. I think the tendency is to go for broke. All right, I'm going to take my sample and I'm going to get 10,000, 100,000 features, or 500 or 3,000 molecules, whatever it may be. I think the challenge is to put that into perspective for the investigator. If you're a lab that's traditionally done more triple quad based quantitation, targeted assays, I think that the discovery type experiments can be overwhelming with the amount of data that you get. So one of the ways in which we kind of help transition people is to suggest trying these really broad-based targeted assays. So for example, we have a lipidomics assay that's LC-based. It was designed for triple quads in mind. And what it is we created this really wide net. All right, we created theoretical MRMs for all the lipids that we could think of. In fact, right now it's up to 2,500 compounds, and look for them. And in a way, it's like a discovery experiment because we're not sure if they're there or not, but we've created a method so if they're there, we're going to detect them. The nice thing about that is you don't have to go through software to interpret your data. It's what you're looking for, and if you get a peak, then you can go through the normal quantitative process to determine how much is there. The reason that's nice is that what you're looking for. You get data that makes sense because this molecule is absolutely this molecule based on retention time, curated MRMs. And it's going to expand the number of molecules that you're looking at. The coverage is not going to be as great as discovery, but you've got an identity and you've got a quantity, right? Once you process that type of workflow, all of a sudden, I think it allows you to really think about, okay, what am I missing? And that's where discovery then becomes really powerful because now you have a really good solid sense on these 500 or 1,000 compounds. And now adding more really makes things a lot more sense because at the end of the day, no matter what type of discovery experiment you're doing, you really shouldn't publish it until you've manually looked at everything and you say in your mind, yes, I'm convinced that's the molecule I'm looking for. Because this black box software approach is really not tenable, especially in the small molecule omics. For lipidomics, I really can't promote any particular software package that's going to do it correctly the first time. You really have to dig in, adjust parameter settings, validate. Again, the xenotoph instruments are very sensitive and fast. Take that targeted method, put it onto the xenotoph. And we're actually done that with lipid mediators, with metabolomics, and we're working on getting that for lipidomics. It's going to give you a midway point from there to jump off and to do more discovery-based experiments. So go into it slowly and make sure you understand each stage of that process before you really jump into the idea of true discovery, lipidomics, metabolomics, or proteomics.
And finally, can you tell us the initiatives your team at SciEx have to promote transparency and collaboration within the scientific community in this field?
Yeah, there's two things we do. I think probably the more important one is the fact that we are sharing our data. So if I write a technical note and I publish this technical note that shows data, and it, granted, it hasn't gone through a rigorous peer review process, and we try and be as correct as possible. But most academics are going to be somewhat skeptical of any sort of white paper coming from industry. And so what we decided to do is actually take the data that we use to generate that technical note and make it freely available on our website. So you can actually go in, look at the data, and essentially prove it. Are these claims that we're making, do they make sense to you? And it's actually been really popular. We introduced that at ASMS in 2025, and very surprising the number of people who have accessed that data and are looking at it themselves saying, okay, I believe it or I don't believe it. So I think it's that additional step where we're adding a bit of trust for people like, here, this is what we have, and this is where we got these results from, and these are our conclusions. But the other aspect of your question, which was collaborations, is we have a very active thought leader program at SciEx, where and for me, it's the best part of my job. I get to work with labs all over the world, where we work together. They have a scientific problem that they want to try and solve, and one of my roles is, because I do know the instrumentation very well and the workflows that work well in our instrument, to try and connect their problems with our solutions. Because I think that's really what the most important part is. When you collaborate, you need, from our perspective, we have to help provide a solution, not just ideas or turn this dial this way. We need to think of the whole picture. And I think we've been pretty successful in that regard, is to engage with scientists at all levels, not just senior scientists, but also junior level graduate students, and help them understand how the instrument works, but how their science could be, the questions that they generate with their science can be addressed specifically with our instruments.
That's great to hear. Well, thanks again, Paul, for your time with us today and for sharing your knowledge and insights.
Oh, it was a pleasure. Thank you so much.
If you enjoyed this episode, remember to check out our other videos for more expert insights. I'll see you next time.